I have the honor of representing a great number of people who work on this project, a lot more than I do, so, I'll talk about that a little bit more, but as I've said to this group before, I do believe that this represents probably the greatest basin-wide collaboration on a fisheries management effort, and I wholeheartedly have faith that it's actually going to result in the successful reintroduction of deepwater coregonids in Lake Ontario. So to start off with the really good news here, the first deepwater Cisco collected since the early 1980s was taken in a USGS trawl this past year. Bob O'Gorman in the audience was probably the last one to handle a deepwater coregonid, and I think he killed it. And this one's dead too, by the way. So anyway, so even though this fish only survived one winter, it's still great news that we have survival, and we've got some more evidence later in the talk that our stocked fish are actually surviving. So this is I'll have multiple confessions. This is the first slide that I have to say I was wrong. I did not think we'd see a stocked bloater in a bottom trawl for at least 12 years. So I was wrong, and I'm glad. Now, this is the most important part of this. There are a great number of folks who have made this project successful, and it truly is remarkable to me, because when I came to Lake Ontario in 1999, Tom Stewart immediately was trying to get me to join in with him in efforts to get this project up and running. To fast forward, Chris Goddard had the vision to get Secretariat staff, namely Gary Isbell and John Detmers, to really try to push the Lake Committee and others and make this work. So there are some key collaborators in addition to those I've mentioned. Kevin Loftus from Ministry of Natural Resources has committed some very, very talented staff time from White Lake Fish Culture Station. Jim Johnson, Tunison USGS, at the time had George Catola in-house, a noted fish nutritionist and very adept at fish culture. We made great strides very quickly. Never would have happened without the help of Mark Holey from U.S. Fish and Wildlife Service, Green Bay, sending his people out to Lake Michigan in January and February each year to collect eggs. So lots of moving parts here, and really, really great. I'm not going to spend a lot of time on this, but we've been talking for several iterations in fish community objectives about reestablishing deepwater coregonids and trying to diversify the prey fish community in Lake Ontario. The goal was to reestablish self-sustaining population in 25 years, and that was based on a stocking target of 500,000 fall fingerling fish. This was covered very well already by Dr. Muir yesterday, but the take-home message is that among the Great Lakes that have lost members of their flock, right, Dr. Muir? Lake Ontario lost more members of their flock than other lakes. So we're currently, as you've heard, we only have the shallow water form in Lake Ontario, but now confident that we're seeing some survival from our stock bloaters. We're not the only lake committee. We're just out ahead of the pack now, but three other lake committees have developed plans or are in the process of developing plans for reintroduction or restoration of coregonids in some form, and we're very grateful that our partners from Region 5 Fish and Wildlife Service are now entering into this as well. So we're hopefully going to have some new firepower in that endeavor. Just look in the lower right, just to give you some perspective about where eggs are being collected in the open circle, and then where the eggs are being sent via FedEx, sometimes overnight, even when they promise it. Sometimes the eggs go to Burbank, California, and still survive, remarkably. Did not kill all those fish that visited California. Anyway, so the northern dot is White Lake Fish Culture Station, Kevin Loftus' shop in Ontario, and the red dot there is Tunison USGS, Cortland, New York. Just a summary of bloater stocking to date, 2012 year class in the left column. Those were the fish reared at USGS Tunison. The middle column are fish that are raised at MNRF in White Lake, and then the total in the right column. The reason you don't see MNRF from the 2012 year class is because they've been stocking advanced fingerlings. We're stocking fall fingerlings from USGS, whereas we're calling them yearlings, I guess, right? In quotations. So, but the take-home message is 190,000 fish stocked to date. The fish that are stocked by USGS are marked with calcine. That's how we were able to identify that fish in the first slide there. But most notably here, we had a great egg take in 2016, not only from wild brood fish, but also from Kevin's brood stock at White Lake. Another thing that I was wrong about. Said brood stock were going to be a waste of time, and I was wrong again. And then again, I'm grateful I was wrong. So here's the rundown on broodstock numbers by year class at White Lake. I'll talk a little bit more about the hormone study and diet studies that are listed there. Already mentioned we had a great egg take in 2016. One big change that Mark and his staff implemented, we were collecting brood with gill nets and having a lot of issues with bringing those fish up from depth and loss of gametes as they came up through the water column and experienced the pressure changes. We've switched now exclusively to trawlers. So we're working on trawlers dealing with fresher fish, dealing with more fish. Also, I think the abundance of bloaters in Lake Michigan is up now, John, as well. So I think that's helping. And so a record egg take this year. Lots of challenges that you can read in our restoration plan, not the least of which is having humans who are nuts enough to go out on Lake Michigan in February to get eggs. They do present very significant challenges in early life history, especially in the hatchery system, and a lot of those problems have already been solved by USGS and Kevin's folks at White Lake. I'll show you some graphs on that, but we secured, well, next graph. So if you look at the three lines here, these represent survival from eggs to different stages. So the red is, or the yellow, green, I'm sorry. Green is egg to eye up, red is from hatch to the end of early rearing, and the blue is from eye up to early rearing, end of early rearing. But in every case, we've seen about a 600-fold increase in survival from our early efforts, which are better than the efforts we had in our garage at Cape Vincent, where Mike managed to kill everything, but it got the ball rolling. I'm sorry, Mike, I contributed to Mike killing all those fish. I can't blame it. Another thing I was wrong about. So this is another great news story. So a group of us collaborated on a proposal for a regional proposal. For those of you who know Great Lakes Fish and Wildlife Restoration Act, if you have multiple states working on a project that qualifies as a regional project, and you see only maybe 1, 2, 3 of those submissions a year, we were successful in securing money for a 3-year study. First objective to look at how we can try to hit that 500,000 fish target by increasing the availability of eggs through both wild and captive brood stock. And then secondly, because of all the hurdles and challenges in rearing, Kevin's staff are looking at running trials on using different diets and different temperature regimes during different phases of development. I won't get into the details. We'll hopefully have a presentation at this session next year getting into some of the details here, but just to give you some preliminary results, one of the major issues we've had very small mouth parts on these fish when they first begin feeding, and traditional commercial diets just are not working. Additionally, you have to have a very small particle size, and it would float on the water. So George Catola developed a spray method to get the feed to drop so the fish could feed. But otohime is a Japanese diet that was very expensive, extremely difficult to get into Canada. So Kevin's staff are looking at some alternative diets to see how they compare to Otoheme in terms of survival and growth. We'll hear more about that. And hormone induction now. As mentioned, we've got brood stock, so we want to see how well we can, whether we can be successful in getting those fish to actually spawn. And early findings indicate that we can, in fact, induce male and female bloaters with LHRHA, luteinizing hormone, releasing hormone analog. But there's some asynchrony between the males and the females, Kevin, so there's going to be some experiments on cryopreservation of milk in 2016 to try to solve that. This is another very interesting project funded by the commission, I believe. Is that right, Tim?
Most of this is from the Restoration Act.
Okay, that's right. Yes, it is another fish. Yeah, okay. So this is a neat study that I was wrong about again. I think this might be the last thing that I'm going to confess on today. So I said, you won't be able to put a scalpel in front of a bloater without the thing dying. You just show them a scalpel, they're going to roll over and die. And not so. Tim Johnson and his partners have been taking brood stock at White Lake and successfully implanting transmitters and getting the fish to live. They've established an array of acoustic receivers and have stocked fish in the fall of 2015, transmitted fish inside this bounded area in the eastern basin of Lake Ontario that you see there in the lower right. And these fish are moving around. They're either alive or they're in the stomachs of lake trout and moving around. But we'll find that out. But I'm confident. That's right. Yeah. Okay. So I already mentioned cryopreservation. Some of the other things that we're looking forward to. As I said, we'll have an update from Kevin's folks next year on some of the diet and temperature trials. I've already mentioned the inclusion now of Region 5 Fish and Wildlife Service in our planning and future efforts. We're looking forward to proving that those tagged fish are actually alive out there. And we need to do some more work. As I mentioned, Tunison is calcine marking the bloaters that they're stocking, and we're hopeful that we can move something along and get all of our bloaters stocked in the future. And as you'll hear from Dr. Weidel here next, we do have extensive deep water trawling, but if that isn't going to be sufficient, we need to be thinking about other ways that we might assess the survival of the project.